Welcome to Biology at Priyajit, my dear students. Today we begin our journey into the fascinating world of plants, algae, the simplest yet most vital photosynthetic plants on earth, photosynthetic organisms on earth, from microscopic unicellular to giant seaweeds. Algae show a remarkable diversity in their cell structure, thallus organization, and modes of reproduction. They basically play a key role in aquatic ecosystems and have immense economic importance for humans. In this class, we will clearly understand their general characteristic features, explore the evolutionary classification by Lee 2008, and study representative forms like Nostoc, Spirogyra, and Polysiphonia. Let us learn how these humble organisms form the foundation of life in water and the base of many food chains. General characteristic features of algae. Algae are autotrophic in nature. They are chlorophyll bearing photosynthetic organisms. They synthesize their own food using sunlight and carbon dioxide and water. Hence, they form the primary producer in aquatic ecosystem. Habitat. Algae are mostly aquatic. Freshwater, they live in ponds, lakes or rivers such as Spirogyra or Chlamydomonas. Volvox is another example of freshwater or aquatic algae. Marine algae, they live in seas or oceans. Example, Sargassum, Polysiphonia, etc. This is the image showing the presence of Sargassum under the seabed, under the sea. Some are terrestrial, which grow on moist soil, tea bark or rocks. Many of algae live in symbiotic association such as lichen, coral, azolla, and amina. This azolla is a fern, a kind of pteridophyte, and these are anamina algae. They interact, the cyanobacterian algae. They interact with the uh, fern through an symbiotic association. So their thalloid body organization, their plant body is thallus. That means their body is undifferentiated, no roots, no true roots, stems or leaves or vascular tissues are found among them. Their thallus may be unicellular, colonial or filamentous, siphonous or parenchymatous. Level of organization. Their thallus ranges from simple unicellular to large complex seaweeds. There no embryo formation occur after fertilization. So this is thallus organization in algae. Unicellular flagellated motile, Chlamydomonas, motile cenobial or in case of Volvox, non-motile cenobial in case of Cinedesmus, siphonoceous form, Vaucheria are heterotrichous based, that is stereocolonium পাওয়া যায়, parenchymatous folios that is found in alva, dendroid type found in Prashinoclodus, falsely branched that is in Cytonoma, unbranched filament that is found in Eulothrix, and non-motile cocoid found in Chlorella. Range of thallus organization unicellular. Their examples include Chlamydomonas and Chlorella, colonial or Volvox, filamentous Spirogyra Eulothrix, heterotrichous Fritziella, siphonous example Vaucheria, parenchymatous alva or laminaria, highly differentiated alga are Sargassum, Polysiphonia, etc. Algal gel characteristics also include their cell wall composition, which is mainly made up of cellulose microfibrils, may also contain pectin, silica, alginic acid, agar, carrageen, etc. Agar or carrageen is found in red algae or rhodophyceae. Alginic acid is found in brown algae or phaeophyceae. Diatoms contain silica in their cell wall, so their cell wall can be differentiated into primary cell wall and middle lamella. Pigment system. The chlorophyll A is the present in all algae. It is the universal pigment of algae. Accessory pigments include chlorophyll B found in green algae or chlorophyceae. So chlorophyceae has major pigments, chlorophyll A and chlorophyll B. Phaeophyceae or brown algae contain chlorophyll A, chlorophyll C and fucoxanthin. Phycobilins or phycoerythrins or phycocyanin are found in chlorophyll, found in rhodophyceae or red algae along with chlorophyll A and chlorophyll D. So these pigments basically help in efficient absorption of light at different wavelengths. Algae general characteristics also include their reserve food materials, which are stored in the form of starch in case of chlorophyceae or green algae. Floridian starch is found in brown algae, red algae or rhodophyceae, Floridian starch. Laminarin and mannitol is found in phaeophyceae or brown algae. And oils and fats are also found in many forms. So methods of reproduction. Reproduction in algae can be of three types: vegetative reproduction, asexual reproduction, and sexual. Vegetative reproduction includes fragmentation, fission, akinete formation, hormigonia, adventitious thalli. Fragmentation is found in Ulothrix, Spirogyra, etc. Fission includes diatom. Fission example include diatom. Asexual reproduction is completed by means of following pores like zoospores, synzoospore, aplanospore, hypnospore, autospore, and tetraspores. Examples are there. Sexual reproduction, it can be isogamy, anisogamy, and oogamy. Isogamy is found in Spirogyra or Zygonema. Anisogamy is found in C. brownie, and oogamy is found in Volvox or Fucus. Algae general characteristics and features include their cell structure. They are typically eukaryotic cell except cyanophyceae or prokaryote. Their cell wall includes cellulose and pectic substances. They are chloroplast with thylakoids and pyrenoid for starch synthesis. Their nucleus is distinct, which is absent in cyanobacteria. Their vacuoles maintain turgidity and storage. They have flagella in their motile stages, which is whiplash and tin cell type. Life cycle patterns. Algae shows haplontic, which is dominant gametophyte or Chlamydomonas spirogyra. Diplontic life cycle, dominant sporophytic stage, which is found among Fucus. Haplodiplontic, both phases are prominent, which is found among Alva, Polysiphonia, etc. Evolutionary classification, Lee et al. Up to divisions. Classification by Robert Edward Lee, 1918. In 1989, Robert Edward Lee classified or proposed the algae into 4 evolutionary groups, 15 divisions and classes. So group 1 is prokaryotic algae. The prokaryotic algae have an outer plasma membrane enclosing protoplasm containing photosynthetic thylakoids, 70S ribosomes, and DNA fibrils, not enclosed within a separate membrane bound structure or nucleus. So chlorophyll A here is the main photosynthetic pigment. Prokaryotic algae has 2 divisions: cyanophyta or blue-green algae. They have chlorophyll A and phycobiliproteins. Division protochlorophyta or prochlorophytes. They have chlorophyll A and B, and there is no phycobiliproteins. Group 2 is the eukaryotic algae. Eukaryotic algae with chloroplast surrounded only by the 2 membranes of chloroplast envelope. They include 3 divisions: glaucophyta, rhodophyta red algae, and chlorophyta or green algae. Eukaryotic algae with chloroplast surrounded by 1 membrane of chloroplast endoplasmic reticulum. They include also 2 groups: euglenophyta or euglenoid, and dinophyta or dinoflagellates. Group 4, it is also an eukaryotic algae group, where eukaryotic algae with chloroplast surrounded by 2 membranes of chloroplast endoplasmic reticulum. So it has 8 divisions like cryptophyta or cryptophytes, chrysophyta, primnosiophyta, bacillariophyta, xanthophyta, eustigomatophyta, rhapidophyta, and phaeophyta, which is brown algae. So we have to study about the brief account of selected algae. This is the image showing the structure of Nostoc. There is mucilaginous sheath, intercalary heterocyst is there. There are akinets, which are the reproductive structure within the vegetative cells. This is centroplasm and this is chromoplasm. So, characteristic features of Nostoc. Nostoc belongs to cyanobacteria or blue-green algae according to algae classification, which are prokaryotic and photosynthetic in nature. Habitat, they found in freshwater ponds, ditches, moist soil, and also in symbiotic association. Example with Anthoceras, cycas, roots, lichens, etc. Thallus structure, they forms soft, gelatinous, spherical or irregular colonies enclosed in a common mucilaginous sheath. Each colony contains numerous intertwined unbranched filaments or trichome. Cell types: vegetative cells are heterocysts. Vegetative cells include photosynthetic bead-like, which are arranged in chains. Heterocysts are large, thick-walled pale cells specialized for nitrogen fixation. Akinetes are thick-walled resting spore for perennation under unfavorable condition. Pigments, they contain chlorophyll A, phycocyanin, phycoerythrin, and carotenoids, giving blue-green coloration. Nitrogen fixation heterocyst possesses nitrogenous enzyme and fix atmospheric nitrogen into ammonia, enriching the soil fertility. Reproduction by fragmentation of trichome into small pieces called hormogonia, which grow into new filament. Asexual reproduction, no true spore, but akinets germinate to form new filaments when conditions become favorable. Sexual reproduction, that is completely absent in Nostoc. Economic and ecological importance, act as a natural biofertilizer in paddy fields due to nitrogen fixation. It improves soil fertility and reduces the need for chemical in a nitrogen fertilizers. This is the diagram of Spirogyra, which contain pyrenoid, nucleus, cell wall, cytoplasm, mucilaginous sheath, chloroplast is there, this cell membrane, this is vacuole, and cytoplasmic strands. So, systematic position of Spirogyra, they belong to Chlorophyceae or green algae and is typical freshwater algae. Habitat they commonly found in ponds, ditches, slow moving streams and lakes, forming floating green mats. Thallus structure, their thallus is unbranched, filamentous and slippery due to mucilaginous outer layer. Their filament consists of a single row of cylindrical cells arranged end to end. Cell wall, their cell wall is two-layered structure, outer pectose layer which is mucilaginous and inner cellulose layer. Chloroplast, each cell contains one to several ribbon-like spiral chloroplast arranged helically. Chloroplast bear many pyrenoids which are centers of starch synthesis and storage. Nucleus and vacuole. A large central vacuole is present. The nucleus is suspended in the center by cytoplasmic strands. Vegetative reproduction, they occurs by fragmentation of filaments into smaller pieces, each growing into a new filament. Sexual reproduction by conjugation, which is an isogamous type of sexual reproduction. It is two types: calariform conjugation found between adjacent filaments, and lateral conjugation found between cells of the same filament. Zygospore formation, fusion of gametes forms a thick-walled zygospore which undergoes meiosis on germination. Life cycle, they show haplontic life cycle, so vegetative plant body is haploid and the only diploid stage is the zygospore. Brief account of selected algae or Polysiphonia, that is diagram, it is the apical portion of a plant body showing central siphon, pericentral siphon, etc. The systematic position, Polysiphonia belongs to Rhodophyceae red algae, the image showing the red colored, and it is a common marine algae. They are found attached to rocks in the intertidal and subtidal zones of various seas and oceans. Thallus structure, thallus is multicellular, filamentous and profusely branched. Their branching is typically dichotomous or alternate. Internal organization or polysiphonous condition, each segment has a central axis cell surrounded by a definite number of pericentral cells forming a polysiphonous structure. Their pigments contain chlorophyll A and D, phycobilins, giving a red colored appearance. They are reserved food, stores food in form of Floridian starch deposited in the cytoplasm. Vegetative reproduction by fragmentation of the thallus. Their asexual reproduction is performed by transfers formed in the tetrasporangia of the tetrasporophyte. Sexual reproduction include by organ by oogamous. Their male gametes are non-motile spermatia. Female organ is the carpogonium with trichogyne. Life cycle shows a triphagic life cycle: haploid gametophyte, diploid carporophyte, parasitic on female gametophyte, and free living diploid tetrasporophyte producing haploid tetraspores by means of meiotic cell division. So economic importance of algae food value. Many algae are used as human food due to their high nutritional value such as proteins, vitamins, minerals, iodine, etc. Porphyra or nori is widely eaten in Japan and China. Laminaria and sargassum are used as vegetables and in soups. Algae are also used as animal and fish food. Biofertilizers, blue-green algae like Nostoc and Anamina fix atmospheric nitrogen. They enrich soil fertility, especially in paddy fields. They are used in green manuring and sustainable agriculture to reduce chemical fertilizers. Agar production, they are extracted from red algae such as Gelidium, Gracilaria, etc. They are used in microbiological culture media, pharmaceuticals, etc. The food industries include the production of jellies, sweets, ice creams, etc. Alginates, they are obtained from brown algae like Laminaria and Macrocystis. They are used as thickening, stabilizing and emulsifying agents in ice cream, toothpaste, cosmetics, textiles and paper industry. Uh, Number 5, carrageenan. that is extracted from red algae such as cornrus, iris moss that is used in dairy products, chocolates, medicines and syrups as a stabilizer. Medicinal uses: algae are rich in iodine such as laminaria that is used for goiter treatment. Source of vitamins include A, B, C, D, E. Some algae produce antibiotics and antiviral compounds. Their industrial uses include that is they are used in the production of biofuels, bioplastic and wastewater treatment. The diatoms are used as filtering agents and in polishing materials. Research and biotechnology: agar and alginates are essential in tissue culture and genetic research. Algal pigments are used in food coloring and cosmetics. Ecological importance: algae are the primary producers in aquatic ecosystem. They form the base of the aquatic food chain. They release large amounts of oxygen through photosynthesis. Environmental role: they help in carbon dioxide fixation and oxygen balance. They used in sewage treatment and water purification. So we have explored the fascinating world of algae, understanding their characteristic features, cell structure, diverse thallus organization, modes of reproduction, evolutionary classification and their immense ecological and economic importance. From simple forms like Nostoc to complex algae like Polysiphonia, these organisms beautifully demonstrate how plant life become and diversified in aquatic environments. So I hope this chapter has helped you to appreciate the foundational role of algae in plant biology. Keep exploring the wonders of plant biology. Thank you.